7-pass receptors. It is also known as serpentine receptor or G protein coupled receptor. Why it is called G protein coupled receptor? Because this 7-pass receptor have coupling of proteins, and the proteins have attached GDP, guanosine diphosphate. Guanosine diphosphate or guanosine triphosphate makes it G protein receptor, G protein, and it is attached with the 7-pass receptor, so it is called G protein coupled receptor. We have discussed receptors. What is receptor? Receptors are macromolecule to which a ligand attached and shows some biological responses. We have 4 types of receptors. One is surface receptors, second is intracellular receptors, and it is also known as cytoplasmic receptors. One is intranuclear receptors and one is extracellular receptors. Then in surface receptors, for example, this is a cell. In surface receptor we have enzyme linked, mean 1-pass receptors, 1-pass, like insulin receptor. One is 4 to 5-pass receptor, which is mainly nicotinic receptor, and one is 7-pass receptor, which is the most common receptor. Why it is called G protein coupled receptor? And this is, and 12-pass receptor is, 12-pass receptor is not a G protein coupled receptor, because these proteins or these receptors have monomeric peptide chains. Monomeric peptide chain. For example, only alpha, only beta, only gamma. But this 7-pass receptor have trimeric peptide chains or proteins. So it is called coupled because long chains of peptides coupling with each other. And why it is called G? Because it have a side chain of GDP or GTP. So it is called G protein coupled receptor. Why it is called 7-pass receptor? Because it crossing or penetrating the membrane 7 times. And why it is called serpentine receptor? Because its shape is like a snake. So it is called serpentine receptor. Today we will discuss the whole mechanism of action of 7-pass receptor, because 7-pass receptor is the most common receptor in body. About 40 to 50% of drugs are acting on 7-pass receptor, and the remaining 50% are dividing in all the other receptors: the intracellular, extracellular, intranuclear, 1-pass, 12-pass, right? So we will discuss the whole mechanism of 7-pass receptor. We will also discuss the second messengers which are making in this mechanism, and it is mainly called the cell signaling or signal transmission in cell, right? So let's start. Seven-pass receptors are divided into, or G protein coupled receptors are divided into 3 classes. G protein coupled receptors. One is GS, GQ and GI. Why they are classified GS, GQ, GI? This is for G proteins. Those proteins which activates and stimulates some biological responses in cells are called GS and GQ proteins. And the proteins which are inhibitory, which inhibits the processes in cells, are called GI. So the GS and GQ are stimulatory proteins and GI is inhibitory protein, right? Now we will discuss some of the important protein in this mechanism. First, adenylate cyclase or adenyl cyclase. Second is PIP2. Third is IP3. Fourth is DIP. Ag. Fifth is TF protein. Right? All these proteins are included in the mechanism of 7-pass receptor. We will discuss all the mechanisms and all the functions of this protein in detail. Now we will take a cell, for example, seven-pass receptor is surface receptor. Some of the hormones in body like FSH, like growth hormone, like parathyroid hormone, they are acting on 7-pass receptor because they are hydrophilic and larger in size and cannot cross the cell membrane, so they are acting on 7-pass receptor. But in contrast, the testosterone, the estrogen, the progesterone, they are lipid soluble, lipophilic, and they can cross the cell membrane as well as nuclear membrane. And some of the hormones are acting in intracellular receptors and some are in intranuclear receptors. Right? So this is mainly the topic of endocrine system. But in pharmacology, we can block or we can activate these receptors. Pharmacologically, we will discuss the drugs effect on receptor. This is mainly the topic of pharmacodynamics. The main thing in pharmacodynamics is mechanism of action, how the drug acts in body. They are some of the acts are stimulatory, some are inhibitory, some are competitive inhibitory, some are partial inhibitory, partial stimulatory, right? So this mechanism is wholly solely for pharmacodynamics of drugs because more drugs are acting on surface receptors. For example, I have a receptor here. This area is called the ligand binding domain, and this is the peptide chain of receptor. Second, fourth, fifth, sixth, seventh. But in normal condition, when nothing is attached with the ligand binding site, these receptors are in inactivated form. Inactivated, right? And some of the proteins are attached with this receptor. For example, this is alpha unit of G protein, and this is gamma unit of beta unit of G protein, and this is gamma unit of G protein. This whole structure is called G protein. This is receptor and they are coupling, so it is called G protein coupled receptor, right? And GDP is attached with this alpha unit. And remember, this is very important for your MCQs, that in inactivated form, in off form, the alpha unit will be attached to GDP, guanosine diphosphate. In activated form, this GDP will be converted into GTP, guanosine triphosphate. These are two MCQs, right? So this is in inactivated form. In GDP, it is in off form. In neighbor cell membrane, we have a very important protein here. And this protein is called adenylate cyclase or adenyl cyclase. And it is represented by AC. right? But it is a very special cutting site, cutting teeth, or cutting processes, which can cut the GTP from the alpha unit, right? So this is, for example, this is the extra peptide chain of this adenyl cyclase, right? Now, for example, some of the ligand, this receptor is GS receptor, stimulatory GS receptor. After GS we will discuss GQ, and after GQ we will discuss GI. For example, here is a ligand. The ligand may be, it may be hormone, it may be drug, it may be neurotransmitter, it may be other chemical substance, right? But in pharmacology we will focus on drugs. For example, morphine. Morphine is very strong opioid analgesic, right, and act on kappa and delta receptors. Kappa and delta receptors are also G7-pass receptor, G protein coupled receptor. So for example, one of the ligand came and attached here. For example, this is a ligand attached to ligand binding site. After this attachment, this protein will be activated, this peptide chain will be activated, and for example, this is the off form of this receptor, and this is the activated form of this receptor, right? When this receptor activated, all we know that here ATP is present in very high amount, ATP, because Krebs cycle is running in cytoplasm, right? So for example, by activation of this receptor, ATP converted into ADP, and one inorganic phosphate react with GDP, and this side become this is the first step, the attachment of ligand with receptor. The activation is the second step. The third step is the breakdown of G proteins. Breakdown of G protein. Breakdown mean that these coupling will be separated, right? For example, the alpha, this is the beta unit, and this is the gamma unit, and the alpha unit, this GDP converted into, this is the alpha unit of G protein, and this GDP is converted into GTP, and now in GTP this is on, it is activated form, right? But here this protein, adenylate cyclase, it will cut this GTP from alpha unit of G protein. For example, this is the activation of adenylate cyclase, and why it is attached, or why it want to cut this GTP from alpha unit, because this adenyl cyclase is responsible for making second messengers, and the second messenger is responsible for biological changes or biological reactions or biological responses inside the cell, right? So this is the third step, and this is the fourth step. In fourth step, this GTP is cutting from alpha unit. So for example, here ADP is present, and this ADP take the inorganic phosphate from GTP and converts it into ATP, and this GTP is again converted into GDP. For example, this is the this is the alpha unit, and the GTP is converted again into GDP. Now these peptide chains in this protein, it will again react, and when react it will form the G protein coupled structure again, and will attach with this receptor and will be waiting for other ligand, and this is cycle. When ligand is attaching here, these units will be separated, it will transfer this ATP, this energy into adenylate cyclase, and it will convert again into inactivated form and will attach to this receptor again for other ligand, right? For example, we are taking a medicines again and again in high doses. A time reached when this receptor becomes saturated and this cycle will be stopped, and when this cycle stopped, the drug is now no more action in body, right? So here the cycle is remain, but here this ATP, it converts into CAMP by the inorganic phosphate which is coming from GTP, right? So this is the mainly this is the second messenger, and all the functions, all the other biological responses occurring inside the cells are dependent on second messenger. How? This CAMP, it is responsible for activation of a very special protein called phosphokinase A. Shortly, it is abbreviated as pKa. What is kinase enzyme? All the kinases enzymes are responsible for phosphorylation. The hexokinases, fructokinases, glucokinases, all are responsible for phosphorylation. So this pKa is responsible for phosphorylation of many proteins inside the cell. For example, this is the fifth step. This phosphokinase A enzyme phosphorylates other important proteins in body or in cell. Right, this is the main function of phosphokinase A, which has been activated by CMP, and CMP has been formed by this GTP, and this GTP is coming from alpha unit, and this alpha unit is activated by this ligand, right? First function, it will activate the kinase enzyme of metabolism. It will activate the kinase enzyme of metabolism, like fructokinase, like glucokinase, other kinase enzyme, hexokinases, and will start glycolysis. What is glycolysis? The splitting of glucose into pyruvate. This is the first change occur in cell due to the ligand. The second change, it also phosphorylate the surface channels. For example, this is the sodium channel. It will phosphorylate this channel and sodium will rush into inside the cell, and this process is responsible for depolarization of cell, right? Next, this phosphokinase is responsible, for example, here we have nucleus, and in nucleus we have DNA. And in cytoplasm, here we have a special type of receptor which is called transcription factor. So for example, this is the transcription factor. Shortly it is called TF. This phosphokinase enzyme also phosphorylate this transcription factor, and when this transcription factor is activated, it will cross this membrane and will come into nucleoplasm and will express a special area of DNA and will form messenger RNA according to the message from ligand, right? So messenger RNA will leave the nucleus, and here we have ribosome, and this ribosome will form new proteins. This is the third change due to this ligand, right? The next, it also activates the proteins which are responsible for proliferation. For example, it activates interphase, and when interphase is activated, the cell will be divided. So it also enhances mitosis. Now listen carefully. If a cell proliferates abnormally, it is called cancer, and when we stop or block this pathway, the cell will stop mitosis, and when mitosis is stopped, the cancer will be depressed, right? So these receptors are important for drugs mechanism, right? So the other changes, so many changes occur due to this phosphokinase. For example, the release of calcium for endoplasmic reticulum, from endoplasmic reticulum, the outing of potassium ions from cell, right? The Krebs cycle, all other metabolic activities, they all are activated by this pathway, and this pathway is called GS pathway, or G protein coupled receptor stimulatory pathway, right? So this is the first receptor we have discussed in 7-pass receptor. The second receptor we are going to discuss is GQ receptor. For example, this is the ligand binding site, and this is the first 2, 3, 4, 5, 6, this is the receptor. These all proteins are called GS proteins, and the proteins which are binding here with this GQ receptor are called GQ proteins. They are called GQ. For example, this is the alpha unit of GQ protein. This is the beta unit of GQ protein, and this is the gamma unit of GQ protein, right? And GDP is attached with alpha unit, right? But the mechanism of this receptor is different from GS, because we have a very special enzyme present in this cell membrane called phospholipase. Phospholipase A is responsible for converting the phospholipids into arachidonic acid, and arachidonic acids further form the prostaglandin in body. But here we have a enzyme or protein called phospholipase C, and phospholipase C is will be activated by this receptor, and when phospholipase C is activated, it will activate a neighboring protein called PIP2, phosphatidyl inositol diphosphate, right? And this PIP2 is responsible for activation of two other second messengers, right? So for example, here I have a very special protein, and this protein have a side chain, and this side chain is attached with other protein here. This protein is called phospholipase C, and this is called PIP2. This PIP2 stands for phosphatidyl inositol diaphosphate, right? Mean that this PIP2 is dependent upon the activation of this phospholipase C, and this phospholipase C will be activated when it gets the GTP from the alpha unit. So for example, here we have a ligand. This is a ligand, and this receptor is GQ receptor. By attachment of this ligand with this ligand binding site, this receptor will be activated, and with this activated, this coupling protein will separated, and when they separated, this beta and gamma, for example, are separated from this alpha unit. This is the alpha unit, and this ATP is break down into ADP, and this GDP is converted into GTP, right? This phospholipase C, when this alpha unit is activated, this protein takes the GTP from the inorganic phosphate from alpha unit and convert it again into GDP. This unit and alpha and beta and gamma unit, it will react again, and when will come back here and will wait for other ligand. This phospholipase C is activated by GTP, and by activation it will activate the phosphatidyl inositol diaphosphate, and this phosphatidyl inositol diaphosphate is responsible for activation of 2-second messengers. One is called DAG, diacyl glycerol, and the second is called IP3. IP3 stands for inositol 1,4,5 triaphosphate. All the DAG and IP3 are second messenger. What is the role of DAG? Diacyl glycerol further activates an enzyme which is called phosphokinase C. It is called phosphokinase C, and when kinase enzyme is present, it will phosphorylate other enzymes and will work. So the first function of this PKC is it will activates different types of proteins kinases, and this protein kinases will activate all these functions, but this pathway is different, right? The next function of this, it also activates, for example, here this phosphokinase, it activates the surface calcium channels, which are very important for muscles contraction. So calcium will rush into the cell. The second, it also activates the chloride channels. This is for depolarization, this is for repolarization, right? These are the functions which are performed by this DAG directly, DAG directly, right? This IP3, this is specially responsible for the releasing of calcium from endoplasmic or sarcoplasmic reticulum, right? This, for example, this is nucleus, and after nucleus we have rough endoplasmic reticulum and smooth endoplasmic reticulum, and the smooth endoplasmic reticulum is responsible for storage of calcium. So for example, these are the calcium stored in smooth endoplasmic reticulum, and this smooth endoplasmic reticulum have a very special type of channels which are responsible for releasing of calcium. So this IP3 activates these channels, calcium channels, and calcium is released from endoplasmic or sarcoplasmic reticulum. Now, what will the calcium do? For example, we have actin and myosin here. This is muscle cell, for example. We have, these are actin and myosin. This calcium is responsible for attachment of actin and myosin and will start the muscles contraction, right? If this cell is neuronal cell, neuron, for example, the cell is neuron, and neuron have a special type of vesicles, and these vesicles stores neurotransmitters and some special types of proteins which are called V snares. Do you remember V snares? And this membrane have T snares protein. This calcium is responsible for attachment of T snares and V snares and will perform the exocytosis of neurotransmitters in synapse. So these are the functions of IP3, inositol 1,4,5 triphosphate. This calcium can also react with or attach with calmodulin proteins, and these calmodulin proteins are responsible for activation of phosphorylations or kinases enzymes, and when these enzymes are activated, it will perform these changes in cell. So this is the mechanism of G chemoreceptor, right? But one very important point here, that after formation of these second messengers, the phospholipase, the PIP2 and adenylate cyclase, how they deactivated. For example, when it forms the CMP, this is the second messenger, it remains activated. GI. Anyone else? Mehran. No, it cannot. GI cannot deactivate this protein. GI can deactivate this second messenger. What do you think? How they will be deactivated? If it remains activated, it will form some issues in cell. It will superactivate the cell. I have discussed this in cell membrane. The phosphodiesterase enzymes. Phosphodiesterase enzyme. In cell membrane we have for example, this is the phosphodiesterase enzymes are proteins. This protein is responsible for deactivation of adenylate cyclase, phospholipase, and when phospholipase C is deactivated, this PIP2 automatically deactivated. So phosphodiesterase, for example, after all these mechanisms, for example, I have a cell, this is a cell, right, and this is a 7-pass receptor, and due to the activation of this receptor, this adenyl cyclase is activated, but when it forms the second messenger, it remains activated, and phosphodiesterase enzyme is responsible for deactivation of phosphodiesterase enzyme PDE. This is responsible for deactivation of adenylate cyclase, phospholipase C, and PIP2. Right? This is so simple. Now we will discuss the GI pathway. GI pathway, very simple. GI pathway is very simple because it will block this second formation of these second messengers. Right? So now we will go for the mechanism of GI receptor, the 7-pass receptor, which is inhibitory receptor. Like for example, this is a receptor. 4, 5, 6, 7. This is GI receptor, and for example, this is an ligand attached to this receptor, and these are the alpha, beta, and gamma. Alpha, beta, and gamma are separated. They are separated. It converts into GTP and alpha, beta, and gamma. Right? This energy is now used for deactivating the CAMP, the IP3 and DAG. So for example, this one pathway ATP is converted into CAMP, and this is the pathway of GS. The second is phospholipase PIP2. It is converted into IP3 and DAG. Right? This is the pathway of GQ receptor. This energy is now consuming in blockage of these pathways, and when these pathways are stopped or blocked, no responses will occur in further. Right? So we can inhibit all these pathway by activation of GI pathway. Now what is agonist and what is antagonist? What is agonist and what is antagonist? All the ligands, all the chemical substances, all the hormones or neurotransmitters which have the ability to activate this receptor is called agonist. And all the chemical substances, ligands, hormones, neurotransmitters, all other things which have the ability to deactivate this pathway is called antagonist. One antagonist is, for example, partially deactivate this pathway is called partial antagonist. If the antagonist completely blocks this pathway, it is called full antagonist. Right? But here all the things which can bind here and activate this GI receptor to block this pathway is called agonist, and all those ligands which have the ability to bind here and block this pathway to activate all the GS and GQ pathways are called antagonist. Antidotes are used as antagonist. Right? The inhibitory chemical substances for the agonist are called antidotes. For example, morphine. If opioid receptors are overstimulated by the use of high dose of morphines, we can stop it with naloxone. Naloxone is the antidote for morphine. Like this is the antagonist, this is the agonist, and this is the antagonist, right? So question you gives. This is your last topic in pharmacodynamics. After this receptor mechanism, we will go for the autonomic nervous system pharmacology.